The Immobazyme team came with their particular construct, a genetically modified organism, the E. coli strain, to produce a particular protein, which is the fibroblast growth factor, and this goes for the meat cultivation application. It's a huge achievement for the CSIR because it will not only establish what we can do now, but for future technologies, it now creates this platform for lab-grown meat applications.
Cell-cultivated meat companies, they take a cell from a cow or a fish or a chicken and they take it to the lab and then they start making that one cell become many cells, and eventually those many cells become a food product. So this product is a growth factor that they use to basically create lab-grown meat. So the burger that's grown in the lab, this FGF2 signals to the cells to multiply and proliferate and become the burger. So FGF2, fibroblast growth factor 2, is a growth factor that is used in the mammalian cell culture industry as well as in academic spaces too. A couple of years ago, we identified that these growth factors were extremely valuable, especially in the cultivated meat industry. So we started wanting to produce these growth factors, and we started with FGF2, but we've since then produced maybe 10 to 15 more growth factors. We've made a protein, it has a specific activity, and it works. This now is not a commercially viable product yet.
They came with the technology for us as a CSR to look at development and scalability. And their main problem was looking at a cost-effective and efficient process. First unit operation is to establish sterility within the culture before we transfer it to the bioreactor. We start there with the microbiology aspect because we want to ensure that we're growing one thing. So when we harvest the bioreactor, that whole broth then goes through a separation process in the downstream process. And we separate supernatant from the biomass. Intracellularly is where our product is. So we have to pop those cells open, to break it open in a process of mechanical disruption. And that homogenization process happens in our pilot plant. Thereafter, we centrifuge to create a clarified lysate, which then goes for a purification process. For the first time in South Africa, we were able to take a process at lab scale to now a 50-liter bioreactor scale and to produce a final product, which was fibroblast growth factor, FGF.
So the BIDC is an incredible facility. It's a world-class facility, but I would almost say beyond the facility itself, the equipment, the machinery, all of that, there was this flow of ideas and it was a collaborative effort, and I think we gained so much insight, industrial insight from them that was completely invaluable. The future holds larger scale manufacturing for FGF, but as well as other proteins and products that we have in the pipeline. We're still at quite a small fermentation capacity, whereas the BIDC has large fermentation capacity, and we're really excited to use that. At the end of the day, we've got a commercially viable product through this relationship, which is really cool to see.
So the fact that they took a unique and new construct that they had developed and produced this FGF in a cost-effective manner, it puts them in a market competitiveness in comparison to what's available internationally. To the CSIR, this opens doors to other novel and unique technologies to localize, particularly fibroblast growth factors and insulin growth factors. I mean, we're competing with products that are predominantly only available internationally, and the cost of production and the cost of manufacture was greatly reduced in these types of technologies that we developed with the Immobazyme guys.